had in the morning, so that they can keep us alert and interest us. So so as Dr. Kitonya has explained, we have the presenters. We're going to start with Samuel Mwendwa. Mr. Samuel Mwendwa is from the University of Nairobi, Department of
Recording
Land
in
Resource
progress.
Management. He's a soil scientist specializing in pedology, the soil survey and mapping. And his current interests are, and expertise is in coding, data analysis using R, and also GIS. So he's also been my student, so I know he's very capable. So welcome, Samuel. Take the floor.
So, good afternoon, everyone. I want first to learn how to operate this thing. Okay. Thank you. So, I'm Sam Mwendwa. I'm a soil scientist. So, I'm presenting on behalf of Sonny Liston, Felix, Grace. We are together, but I'm the one presenting. So if I had not put Ocheng after Sonny Liston, you would probably guess the next name. So Ocheng is not here, he's absent today, but I'm with Felix here today. So our topic is geoprecision. So I was in Form 1 almost 20 years ago, and the geography teacher said, geo is indicative of earth or soil. Then, of course, we are here for precision agriculture. So, harnessing geospatial data and the geographical detector model for smart soil nutrient management, and ours is an exposition. So, for some background, So, for some background, we were motivated by the capability of precision agriculture having transformed modern farming using data-driven decision-making. And if you follow our presentation, you'll find that if you just read where we have highlighted in red or where we have written in red, you can make some point, some sentence or something. So the aim is to present an approach that synergizes data collection, soil analysis, mapping, and of course, I know maybe for most of you, I'm introducing the OPGD model, which is the optimal parameters-based geographical detector model to enhance site-specific nutrient management. So we have an application which is online on Play Store, for those of you who are on Android, called SW Maps. So it has a questionnaire, a photo capability, and you can also do GPS. So you can just fill your data on that questionnaire, then you collect data, then you take a photo of your study area, then you take the GPS coordinate. Then you do the mapping in QGIS. I usually use QGIS because, of course, it is open access. Then, of course, the OPGD model is in R. Then the trick is usually very simple. So you systematically gather soil samples from various field zones. Then, of course, you go to the lab, do analysis about the physical and the chemical properties. Then, when you go to do the data collection, you may decide to follow some protocol. For example, we have some protocol called stratified random sampling, whereby when you go to the field, you can delineate the field based on, for example, you have heard that I'm a pedologist. Let's say, for example, the 5 factors of soil genesis. Maybe the slope is not uniform. So you decide to take some samples at the slope where maybe it is more than 30%, then maybe 10 to 20%, then on the flat side, so that you can attribute soil differences to the slope, or probably sometimes the land use. For example, the vegetation. You don't expect soil samples taken from a bare land to have the same organic carbon with soils that have been taken from the forest. So that's why I'm talking about a protocol, so that you also don't fail to make informed decisions about the heterogeneity. So to maximize the impact of nutrient application, that's now where the model comes in. So it is going to give you the key factors influencing differences, which we call spatial heterogeneity. Let's say, for example, the factors influencing crop yield. We are going to use crop yield for our example. So the model just provides a powerful framework for quantifying the influence of individual factors. I'm going to show you that, not practically, but I got screenshots to show you what I usually do. Then, when you are using this model, you want to uncover insights into the most significant drivers influencing your response variable. I'm going to show you what I mean by the response variable and also the factors. So, the model has a detector called interaction detector. That is going to show you how combinations of factors affect our example, which in this case we are talking about yield outcomes. So, based on the findings you uncover, you can now talk about precise nutrient application. So, when you integrate this model, the farmers can have some scalable method, and I'm going to show you how this technique goes to the farmers for managing field variability, and of course, ultimately, you are going to enhance profitability. And above that, environmental stewardship. You are going to conserve the environment because, you know, when you apply just what is needed by the crops, you won't have issues, for example, eutrophication or pollution of water elsewhere. So, previously, studies have demonstrated the immense potential of merging precision agriculture and spatial data analysis to create site-specific management strategies. So, you are going now to have smarter, more sustainable practices anytime you decide to go the data way. You are informed by data about what you put where and when not to put anything on your land. So this exposition is going to present to you the importance of embracing advanced technologies and data analytics for sustainable agricultural development. So, spatial variability of soil properties. This one I won't dwell much on this. I want to go to the model because many people have talked about this, and this is just straightforward. Yes. So, it's just about the problem is just low soil fertility, inadequate soil info, and of course climate change that threaten productivity. Then we are working towards sustainable development goal number 15 on soil health. So to achieve that, you need now to tailor your applications based on soil properties. Geostatistical techniques can effectively assess this variability. And anytime you have soil samples, at least 30 soil samples, you can be able to perform geostatistical techniques. Then the other issue is usually the blanket fertilizer application, which disregards the heterogeneity, and that one will always lead to suboptimal yields. Those of you who are in agricultural economics must have heard about the Bosserupian theory. So increasing population usually demands innovativeness, and that is why you are here. So understanding soil variability is essential in several applications. So I'm going straight to the model, and then I show you these applications. So, for example, I have not supervised but co-supervised 3 undergraduate students to publish their work in peer-reviewed journals. So the point is, when you want to do some precision agriculture thing, is usually not anything complicated. This is from one of my publications, but not with a student. I was with Professor Kironchi. This is part of my master's, one of my objectives. So you can see that I had an aim, I had a farm, then I went to the lab, I did data analysis, I have my key findings there that the soil properties varied, especially in the farm, and then I have a recommendation. So, however small your farm is, you can always do precision farming. Then, it's just, and I won't dwell much on this because this has been said, and I have also said most of it, but when you just read the REDS, innovative farming management concept, and it is going to utilize advanced technologies. So, of course, it will involve the use of the GIS, data analysis. The result is high degree of accuracy in farm management. So, the field data, of course, I had talked about the SW maps, which has a questionnaire, photo, and GPS capability. So anytime you are doing variable rate technology to apply inputs at varying rates, you are going to protect ecosystems. I told you to follow the RAIDs. Then we have now the geographical detector model. So this is just a screenshot from R. When you look at the very first sentence, not sentence, but line of code after the two comments, you are going to find that I'm installing packages. I'm installing a package called the GD, and of course with the dependencies, true. Then, of course, I load the library. Then I'm creating an object called sugar GDM. Then the function is called GDM. So, for example, for this one, I was working for a client. So you can see, I want to see what is influencing the CEC the most. Is it the landform, the microrelief, the land use, or those other soil properties? Then you can see I have continuous variables. What I'm doing to those continuous variables is called discretization. I'm making them discrete. On the right side, of course, is a screenshot showing that it is the calcium that influences the CEC, in this case, the most, the one in the red. And what is on the right side is called the geographical, it is called the detector variable. So I'm going to show you in the next few slides about that. So the GD is just a statistical method to analyze spatial heterogeneity. And it was published by Song et al. 2020. Anytime you just go to your Google and search Song et al. 2020 OPGD, you are going to come across this model. Then you find that previous methods were just subjective in criteria, so they did not quantify anything. So you find that the OPGD model addresses that gap because it optimizes parameters allowing for more accurate extraction of geographical characteristics from diverse data types. I'm going to give you 3 examples of data types for application in this. So it is going to enhance the flexibility and effectiveness of spatial analysis. So, for example, the geographical detector model usually consists of 5 key components. And this one you run in R. So the factor detector is here. This is from now Song et al. 2020. I'll show you the study in the next few slides. So you find that in whatever he was trying to check the influence, you find that precipitation played the most important role in that because it is the topmost, the one highlighted in red. Then we have parameters optimization. Then we have an interaction detector, the risk detector, and ecological detector in the following few slides. So the factor detector, this one here, it quantifies the relative importance of explanatory variables using the Q statistics. Q statistics is just synonymous with the F statistics for those of you who are good in stat. It's just like the F stat, variability between versus variability within. Then you have parameter optimization, which involves selecting the best methods for spatial discretization. And that is why now I was telling you, if you have continuous variables, you have to make them discrete. Then this is the interaction detector. It is going to tell you now whether interaction between any two of your variables is enhancing the CEC, for example, in our case, or it is weakening the CEC. And then you'll see there is some legend there. So that legend is telling you small dot, small. Medium dot, just medium influence. Large dot, it means that that one is influencing it much. And then you can see the color there. This one comes automatically. So it is enhancing the CEC, or it is enhancing, this is nonlinear, by I can't recall the correct name at fingertips. Then the other one is weakening. Anytime you see the blue, it is weakening. So it means that any interaction between these two is weakening. So it means that the CEC is going down. The risk detector usually appears like this. This is just a screenshot for the pH only. So anytime you have the pH, the copper, or soil properties, each will have this. So you can see for this one, what it's telling you is that where you have the red, that is where the most CEC is. And I think for those of you who are soil scientists, that makes sense. Because you know, as pH drops, most micronutrient cations are usually toxic. And then the blue one means now, that is now when the CEC will be going down, the blue one. Then the red one going high. So the risk detector evaluates whether the mean values across different subregions differ significantly. Then you have this ecological detector in just a few minutes. This ecological detector. So this one, it just tests the significance of the influence of one exploratory variable over the other. So you can see these are just yes, no. Is this one, the interaction between this and this affecting CEC in any way? Either yes or no. These ones will just generate automatically the moment you run it. So in Song et al. 2020, you are going to find these three cases. So you'll have the first case, the second case, the third case. I'm encouraging you to go and read it. You'll see all those cases because they are using different types of data. This is the process. So I'm also encouraging you to go and read the process. By the way, I'm giving you this immediately after this presentation. So this is how the discretization occurs, the factor detector, everything up to the ecological detector. So this is just the OPGD process. Then after now you do the OPGD, you have what we call spatial autocorrelation. What you want to know is if higher values of CEC are dispersed or accumulated within the same zone. Because, for example, and by the way, you can see this is just Kisumu. If you look at these dots properly, if you look at those dots properly, and because that is like Victoria, you'll find it is in Kisumu. So the importance of this spatial autocorrelation, you use a package called Leaflet in R to have this map, so that you can know where that dot exactly is. So where you find more red dots, you know that is high, bordering high. Where you find purple ones, you find that is low, bordering high. Then the rest are all low. So you can now see, for example, this area is high, bordering high. It means that the CEC in that area, and you can see the area specifically, it has high CEC, so you don't need to put a lot of remedies on that. So for our conclusions now, we are encouraging integration of the OPG model and PA for improving nutrient management. Then adopt advanced technologies. Then continuous research. But you see, farmers, smallholder farmers, do not understand these models. So this is our message to you as researchers. When you publish your paper using these techniques in high impact journals, now it is your responsibility to go to the village, call your chief, ask for a baraza. Then you can just call 20 people to that baraza. You can promise them meat and eggs, so they will come. So tell them to draw a map of the village in 1971, the way it looked like on a manila paper. Then tell them again to draw the same map of the village the way it looked in 1991 after 20 years. Then another one for 2021, that is after 30 or 20 years. So go back to Nairobi, digitize those maps, and ask for another baraza now with a lot of people from the village. Tell those 20 who drew the maps, are you the ones who drew these maps? Because we have digitized them. They will say yes, then tell them whether they are seeing any changes. They will tell you now we had 4 rivers in 1971. In 1991, we had 2 rivers. In 2021, we have nothing. We had a forest in 1971. 1991, we have nothing. In 2021, we have nothing too. So you are going to tell them whether they are seeing any problem. They will start looking at each other. Then they are going to make recommendations for themselves. So that is how you can pass these kind of complicated technologies to the farmers. And for that, if you want this, please scan that QR code to have that presentation. And I'm giving you 10 seconds. 10 seconds. Just scan it, you'll have that PPT. Professor, just give them 10 seconds. Yes? Yes? Oh, no, the chief's baraza is that village gathering by the local. Thank you so much. We are telling you to challenge the future. Okay, that is well noted. Okay, that is well noted. Thank you.
Yeah, if we don't have another, then let's give a clap to Samuel. So thank you, Samuel.
Thank you.
Then we'll read your report. The next.
Exploring the potential. Come on.
Our next speaker, do you have Dr. Kamau? Is it uploaded? The team uploading? Yeah, that's Dr. Kamau's presentation. Can introduce him. Dr. Solomon Kamau is also a soil scientist. He was formerly in the University of Nairobi. He's more into soil fertility, as you can see his title, and he's been working on issues on biochar. So his title is Exploring the Potential of Biochar and Microbial Inoculants for Sugarcane Production. So that one I missed out, I'm sorry. You can please enhance the introduction. Yeah, thank you, Dr. I think now it's in Moi University. I had missed that out, I'm sorry. So, Dr. Kamau, proceed.
Okay, good afternoon, everyone. My name is Solomon, and as Prof has introduced me, this is my alma mater, University of Nairobi. I was based in Kabete, but now working with Moi University as a soil scientist, and my interest more towards soil biodiversity, soil fertility, and issues, especially issues on biochar. And my talk today is more of what we are trying to do and see how far we can go with it. Mine is not so much into precision agriculture, but it's more of one of the components within the larger issue on precision, because we are looking at issues on fertility and how we can try to manage, for instance, the losses of soil nutrients and how that can reduce impacts, issues on runoff, and how we can retain most of the nutrient within the soil. Currently, we are working with the Sugar Board, trying to see how much we can, or we can try to come up with a product, a fertilizer product that is tailor-made specifically for the sugarcane sector. So just an introduction. I'm working with Professor Nancy Karanja and other team members from Sugar Board. Professor Karanja is also a soil scientist. She's now retired, but she was based in Kabete. And still we are working with her on issues of biochar. And we are going to introduce more issues on biochar later on in the presentation. So if you look at the sugarcane sector, it's being affected mainly by the reduction in the fertility within the sugarcane belt. And if you look at mostly the soils that are dominant within the sugarcane belt, mainly acrisols, and ferrasols, but still we have pockets of other soil types like vertisols, especially within the Morhoni areas. But these soils are inherently deficient of most of the nutrients, and therefore there is a need to try to come up with a product that will retain as much nutrients as possible, because these soils have been cultivated for so long, and therefore even the issues on fertilizer recipients, when the fertilizer are applied, sometimes the response is not so good because issues on, for instance, the soil organic matter content is decreasing, and therefore when you do apply these fertilizers, especially the inorganics, they tend to have lower response rates. So if you look at the sugarcane production rates over time, you can look at those three lines. You have the ultimate production, you have the area being cultivated over time, and then the production per unit area. So you can see the production, the tonnage is increasing because it's mainly being driven by the increase in the area that is being brought into cultivation for the sugarcane. So this is not a reflection of the true reflection of the production per unit area. So if you look at now the third graph in red, that is the production per unit area. So you can see it's decreasing over time. And if you look at, for instance, the production in 2020, the national cane yield was around 64 tons per hectare. That is the average, compared to around 85 tons per hectare that was being realized in 2001, between 2000 and 2010. So meaning, even if the area under cultivation is increasing, but the production per unit area is still declining. And if we were to stop increasing the amount of land that is being brought into cultivation, it means then you have a very big drop in terms of the production. And therefore, there is a need to address this issue. So you see, in a period of 20 years, there is a decline, 10 years, sorry, there is a decline of over 20 tons per hectare. That tells you there is a lot that needs to be done. But of interest, our interest is mainly on the waste that is coming from this sugar sector. And most of it is bagasse and filter mud. That makes the biggest chunk of waste that's coming from the sugarcane industry. And if you look at the ratio of the input, that is the sugarcane, and the output of the waste, there is, if you were to take in 10 tons of sugarcane, there is about 10 tons of bagasse that is coming out, and about 0.3 tons of filter mud, or what we call the press mud. And the estimates, based on the estimates, there is about 2.4 million tons of bagasse that is being released yearly, and about 240,000 tons of press mud. And there is also additional waste that is retained in the farms, about 1.5 million tons of the cane tops, whatever is harvested and left in the farm. And if you look at the amount that is being used yearly, out of the 2.4 million tons of bagasse, only about 25% is being utilized within the factories and sometimes outside of the factory. And therefore, there is still a significant amount that is left unutilized. So meaning if the amount, if you look at the estimates of the contents, that is the chemical nutrients within these two waste, you can see a filter or press mud. It has significant amount of phosphorus as well as potassium and calcium, the ones highlighted there. So meaning if we are to leave this material unutilized, there is a lot of nutrients that are being wasted. But bagasse, you can see it's quite low in nutrients. And therefore, there is another way then of treating this material, which can then be utilized as a fertilizer. So if we are to do a quick estimate of how much we are losing in terms of the nutrients, so out of the 2.4 millions that are not being utilized, at least 500,000 kilograms of phosphorus is lost, okay, for not utilizing the filter mud or the press mud. And then there is an additional of 200,000 kilos that are being lost when we don't utilize bagasse. So if you are now to equate this in terms of the bags of what we are usually using in Kenya, the DAP is a common fertilizer. So if we are to equate that amount, then we are losing almost 70,000 bags of DAP annually for not recycling these materials. If you look at the N, that is 7.2 million kilograms that are lost on press mud alone, and 3.6 million kilos when we don't utilize bagasse. So meaning if we are to equate the amount of these nutrients in bags of DAP, then we are losing almost 1.2 million bags. So this is a very significant amount of nutrient that is just going into waste. And if we were to utilize, meaning we can cut back on the amount of fertilizer that we can apply in these areas. So our interest then is to see how we can utilize these materials, especially bagasse, because we have said bagasse is quite low in terms of nutrients, and therefore applying it directly may not benefit so much the crops. And therefore converting it to a material that can be or can sustain these nutrients, not directly, but it can be used to sustain the nutrients if it can be combined with other forms of fertilizers, then we can be able to retain as much as possible. Okay, so as an introduction to those who have not heard of bagasse, or sorry, the biochar is mainly a carbonized material. Is a product of pyrolysis in a low oxygen condition. And the product is carbon rich. And this can, apart from harnessing the nutrients within the soil, it can also be used to sequester the carbon. Because when it's converted to biochar, then this material can stay up to millennia without being degraded by the living organisms. So why biochar? Recently, studies have shown that there is an interesting trend in some of nutrients when biochar is applied. Apart from that, there is an issue in terms of incorporation of or enhancing soil biota. And you know soil biota is one of the key components within the soil which drives most of the processes. And of interest is the incorporation of organic matter in the soils, enhancing issues on soil structure, and so on and so forth. So this is one of the components that we are exploring on how we can enhance the soil life using biochar. So apart from application directly to the soil, as I've mentioned, we are looking at the ways of how we can enhance this biochar directly by using it as a carrier of inoculants. And some of the studies, because this work that we are doing is still ongoing, and we have not done so much, but previous studies have shown that if you were to use plant growth promoting bacteria, for instance, you can cut back on the amount of phosphorus that is required by up to 75%. And another 70% nitrogen, meaning that then you require very little amount of fertilizer if you were to enhance the soils using these plant growth promoting bacteria, for instance. So one of the products that has been tested and it has been proved to be working, and actually it has been patented, is Biofix, which is a product of Mea Fertilizer, as well as the University of Nairobi. It's patented. And this is mainly for legumes. Now we are trying to explore if there is a way we can use such a type of a carrier to enhance the production within the sugarcane sector, because this has been proved to be working in legumes. And to start by, because this work was started way long before, this actually was the basis of my PhD. We started by asking ourselves, because there was that interest in terms of application of biochar, but our interest was, before we can apply the biochar in the soil, what is there already that is existing that we could use to inform us on how we progress? And we started by looking at those areas that had been recently converted from the primary forest. This is mainly in Nandi, the Kakamega Nandi Forest Complex. So we looked at those areas that has been cultivated for several years in a chronosequence pattern. That is farms that have been converted recently towards those farms which had been cultivated for quite a long time, that is over 100 years. So there was that chronosequence kind of setup. And of interest is that some of these farms had been converted very recently, as early as 2000. And therefore, the question that we wanted to ask ourselves, because the mode of conversion of these areas was to cut the trees and burn, or rather to prepare charcoal. So that was the first entry point, because biochar and charcoal, the only difference is that the interest in terms of the materials that is being prepared. Charcoal is mainly towards energy. Biochar is specifically for use as a soil amendment. So we wanted to know these spots that has been used to prepare this charcoal, how is the response in terms of the soil biota as well as soil nutrients? And we identified those areas with unknown history and in terms of the age of those charcoal making spots are similar or within the same period of time to try to understand the impacts. And from this, we saw there is a kind of an interesting trend because most of the, if you look at the nutrients that we looked at, most of them were higher within the charcoal making spots and decreased as you move out of the charcoal making spot. So the black ones represent those at the center of the charcoal making spot, meaning that then you have a higher concentration of these nutrients within those charcoal making spots. Checking out the soil biota, our interest was mainly the macrofauna, and you can see the highlighted in yellow, the earthworms, for instance, especially the endozoics, the ones that are mainly taking a lot of soil, you could see there is a decrease, or rather an increase, moving towards outside of the charcoal making spot. Okay? So generally there was a decrease, meaning that there was higher impact of these earthworms when you have a higher concentration of these charcoal fines. So if you want to get more information already, that information is elaborated in the paper, so you could check it out because it's quite comprehensive. The second, after we checked this, we went now further from now, based on the understanding that we had, now moving now to application of this biochar. And this was done in Kabete. This was more of an on-station trial. So, and the biochar was prepared from Prosopis juliflora, which is, it has been termed as a noxious weed. So we wanted to see if then we can utilize this noxious weed to try to prepare this biochar, because our aim is not to cut down trees to make biochar. It is to utilize what is readily available to make the biochar. And you can see, in terms of the response, biochar alone was actually producing more than 5 tons per hectare. Compare that with the control, meaning that you are getting more than 5 times the amount of yield when you apply biochar. And this impact was sustained even after stopping the application of this biochar for another 2 seasons, meaning that it has more impact beyond the application period. Based on that already, again, the publication is out, so if you want to get more information, you can get it from there. And therefore, now, our interest now was based on those studies that we have observed, and now we are trying now to come up with a product that can address the issues that we have mentioned in the sugarcane sector. So basically, this is a summary of how the impact of biochar that can be felt, that in terms of issues on CEC, water retention, nutrient availability, organic matter concentration, issues on soil structure improvement, and enhancement of microbial activities within the soil. So all this then will feed into issues on the nutrient retention, issues on increasing in terms of the productivity of the sugarcane yield. So this work in progress, and when we have come up with the product, perhaps then we can share in the next conference. Thank you very much.
Another round of applause. So we'll allow now the questions or comment to Dr. Kamau. Start with
Sorry, just last week I was in a liming workshop at Caro headquarters, and I was mentioning about voucher, and people were surprised. I was talking about voucher for liming. I'm glad you have done this work. I've not been able to access it. When did you publish it?
That was published in 2017.
How come?
Yeah, it's a long time, but still it's work in progress. But I can share that
Yeah, please.
publication.
I need to share because it's very important.
Yes.
First of all, you have proved a few things that voucher is good for soil fertility improvement. And you have even gone further and showed that Prosopis julifera madenge, as people know it, can be useful. And that's exactly what I was telling them.
Exactly.
Then I was telling them we can also get voucher from sugarcane bagasse. And
Yeah.
all this is in your papers. So I'm
Exactly.
glad you've done this, and I congratulate you for that.
Thank you.
Thank you, Dr. Silaba, for those comments, and we're happy that this work is now coming to being disseminated at this level so that other can see most people here are young, and they can be able to take or join up with Dr. Kamau and take this to the next level of now using precision agriculture. Any other? I don't want to cut anybody out. Any other comment or question, please? Then if we are okay, we can give Dr. Kamau another clap. Thank you, Dr. Kamau. Our next presenter is Victor Kipto. Are you with us, Victor, or is it online, Daktari? Victor, come. I'm introducing him. He's Victor Kipto. He's going to give us a presentation titled Residual Effects of Land Preparation Method and Organic Amendment on Soil, Physical Property, Growth, and Yield of Collards. Before he starts the presentation, just as an introduction to the presenter, Bwana Kemboi is a graduate of Meru University of Science and Technology, and he has an MSc in horticulture. He's also a part-time lecturer in the same institution within the Faculty of Agriculture. And he's passionate, he's got a keen interest in horticulture and restoration of soil health. So, Victor, karibu.
Okay, good afternoon. Yes, this is Victor once again. And thank you for this opportunity to come and present at Africa Conference on Precision Agriculture. Yes, to me, I'm a fresh graduate of Meru University. In fact, 2 months old now. Graduated on 4th October 2024 with a MSc in horticulture at Meru University. And, as we wait for them to open the PowerPoint for us, since we started this conference, we have had many stories and many solutions and innovations that people are coming up with concerning food security, the thing with the main objective of improving the food security and also improving the soil. And to my case is that not only we are talking of yields and yields and yields, but we have also to talk about the health of the foundation of everything, which is the soil. And when our soils are healthy, then our crops will be healthy. And once our crop will be healthy, then even our bodies will also be healthy. And therefore, the soil health is equivalent to crop health, which translates significantly to our body's health. Okay.
ICT? Oh, it's come. Thank you.
Okay, this was part of my master's research work, and previously we were working on the effects. We were working on the effects of land preparation method and organic soil amendment on soil properties, growth and yield of maize. And as we were discussing with Professor Masinda and Dr. Moria, we were asking ourselves, whatever we are applying in our soils as the organic manure, does it have any residual effects on the succeeding crop for crop rotation? And we came up with this idea of checking the residual effects of organic amendment and land preparation method, which was conservational land preparation method, because in our methodology, you will see that we have two land preparation methods. That is, we had a zero tillage, that is in basin-like method, and conventional land preparation method, which is the normal tillage that we have. And for the organic amendments, we were working on the black soldier fly frass fertilizer, because we had a big project on black soldier fly at the university, managing the waste. And the waste from the black soldier fly, we were using it to check if it can help us in growing our crops. Okay, we were using collard, which is a sukuma wiki, because it's a staple food crop here in Kenya. And the reason why we opted for collard is that collard is always, it normally requires high nitrogen because of its vegetative growth and the leaves. And that's why we opted to pick on collard to check the residual effects of organic amendments that we amended in the soil. Allow me to go fast. And also, as we know that there is over-reliance on conventional land preparation method way, that is the normal tillage. And again, there is also over-reliance on inorganic fertilizers. We have heard of people discussing on how much amount of inorganic fertilizers or DAP or CAN we can apply without thinking of how will influence the health or how will influence the population of the microorganisms in the soil, because the health of the soil comprises of the three properties, that is the soil physical, the soil biological, and the soil chemical. And organic manure now is becoming a good source of nutrients for crops, production, and also sustainability. And our problem is that, yes, there is deterioration of soil physical properties in terms of soil moisture retention, decreased soil fertility, and also there is also limited information on the residual effects of organic amendments on the succeeding crop. And to justify what we were doing is that coming up with an appropriate land preparation method will help to increase food productivity in arid and semi-arid regions where they experience low rainfall. Also, use of organic amendment will also improve the crop production as well as improving the soil moisture retention, which will help to increase the production of food in arid and semi-arid regions. Our objective was to determine the residual effects of land preparation method and organic soil amendment on growth and yield of collards. And the research question is that, does the land preparation method and organic soil amendment that we apply has an effect or has a residual effect on the succeeding crop. We did this at Meru University, demonstration farm, which is in a semi-arid area in Meru lower zones. And the experiment, we had two land preparation method, that is the zero tillage, the conservational in basin method, and normal tillage, which is conventional, and the 10 subplots amended with different organic manure levels. That was our treatments, and that was our layout. The first one, that was the basic method, that was zero tillage, but we prepared some basins-like holes. On the only place where we were planting, these plots was for maize experiment, and this one was the crops which was succeeding the maize, as you can see. This was our materials. We were using the rice husk biochar to check their effects also. We were using the black soldier fly manure to check their effects also, and the trichoderma. This one was for maize. And from the results, let me go direct to the results. We subjected our results to SAS for analysis to determine the significance. From the results, we found that organic soil amendment improved the soil physical property. That is, improved soil moisture, and yes, there was increased soil moisture, and also the land preparation method, there was significant influence on the soil moisture retention. And this was the result, and we were using this tension meter to monitor the soil moisture. For soil bulk density, we were also checking the soil bulk density of the soil, and we found that for land preparation method, there was no any significance. But on amendment of organic amendment, the ones with the biochar and 5.5 tons per hectare level of black soldier fly manure, it recorded the lowest soil bulk density compared with the control and the ones which had chemical fertilizers like DAP and CAN. As you can see, the control and the ones with the DAP had a higher soil bulk density of about 1 grams per centimeter cube. For the yields of maize, these are just the brief results for the previous results for maize, as I come to the residual effects now of this organic amendment. And we found that for conservation land preparation, yes, it influenced the maize yield significantly. And the one with conventional, the normal tillage, recorded the lowest, and this might be due to the excess loss of moisture. But for the conservation, the increase could be due to increased soil moisture holding capacity. Same case to the application of organic amendment. For those with the high rates of black soldier fly manure, it recorded the highest yields, which was more or less the same with the DAP and the CN, and the rates which we used here is the normal rates that farmers use. And as you can see that the result for maize was almost the same with the 8.5 tons black soldier fly manure. But for the biochar, we didn't saw any significant impact on the yields. As well for the trichoderma, this one we were using to check their impact. If you amend the trichoderma, which is a biofertilizer, with an organic fertilizer. And we saw that amendment of trichoderma, it influenced the yields of maize. Now, on the residual effects, allow me to explain on the collards greenness, the spud, the chlorophyll, and also the yields of collards. For land preparation method, there was significant influence on the chlorophyll index of collards, as you can see, with the conservation one, recording the highest levels, and the conventional recording the lowest, and maybe is due to the leaching of nutrients. On the amendment of different levels, for the trichoderma, from the previous results, you saw that there was significance in terms of trichoderma, but here, I didn't include here because there was no significance on the residual effect. So I just show you the ones which had a significant residual effects on collard, and that is the application of 8.5 tons BSF, which recorded the highest SPAD readings, or the chlorophyll colors. But for the yields, it's also the ones with the high levels of 8.5 tons black soldier fly recording the highest yields. And this was the cumulative leaf yield. And the conservational one recording also the highest levels. The reason why we use also the farmyard manure in that experiment is that this is the normal organic manure farmers using, that's the cow manure. So we were using what farmers are doing with what we are getting and what we are trying to understand their effects on crop productivity. And as you can see, the one with the chemical fertilizers almost the least. And this could be due to the exhaustion of nutrients by the previous maize, which was maize. But as you can see that from what we got is that application of organic amendment has a significant residual effects on growth and also yields of collards or any other crop that you can use, which is any vegetable crop. That's what I was explaining. And in our conclusion is that increasing moisture in conservation land preparation method, it influences the growth and yield of crops, and this also helps due to the reduced runoff. Again, the results from the second experiment shows that organic amendment not only helps the crop, but also it helps for the long term, but for the chemical use or the chemical fertilizer use helps the crop or the sustained crop for the short term. So it means that the inorganic fertilizers is only useful in the short term, but for the organic amendment helps, or it's useful for the long-term use. Thank you. Yes, this was, to mention on this picture, these were the pictures of the collards which we harvested. The one is from the plots with black soldier fly manure, and as you can see, the root mass for the control, and also the root mass for the leaves for the collards, which was in inorganic fertilizer application. Thank you.
Thank you, Victor. Let's give him another round of applause. Yeah. Any question or comment? Any question? It means his presentation was very clear. So we thank you for the work. Our next presentation is going to be online, and this is going to be by Bernard Mudengi. Bernard. Our next presenter is Mr. Bernard Mudengi Yubia. He's an MSc graduate. He holds an MSc in Agricultural Resource Management from the University of Nairobi. Currently, Bernard works as an agricultural officer in Kangundo sub-county in Machakos, and he has a rich experience of 18 years in extension services. So it's good now we're coming out of the research or academia to the research extension and dissemination person. And this, as we said, is going to be online. And the title, as you can read, is The Effect of Green Manure.